session. So now we'll move into the next generation sequencing technology, that is fourth generation sequencing technologies. Okay. Okay. So now we'll see what are the next fourth generation sequencing technologies. Okay. So fourth generation sequencing technologies is actually nanopore based sequencing. Okay. So we have seen already how sequencing was done using the protein nanopore, that is Oxford nanopore technology. Now we'll see here the fourth generation sequencing technology. Okay. So nanopore based sequencing is actually considered the fourth generation sequencing technology. It's also known as single molecule nanopore technology. Okay. So with nanopore, personalized medicine has actually increased a lot. Now, what do you mean by personalized medicine? Okay. So here, before getting into what is personalized medicine, now what is the medicine what we're getting at this point? Whether it's a generalized medicine or a personalized medicine. Yes. Whether it's a generalized medicine or a personalized medicine, what we're getting at this moment.
Sir, generalized medicine is like for every person, it's the same. Like for malaria, hydroxychloroquine is given for every patient. But
yes,
whereas in personalized medicine, according to the compatibility of compatibility and the particular individual, it will be given, sir.
Yes. So the difference is that generalized medicine. What is that? What we are getting in at this moment? When you go to a doctor, doctor will give you the same medicine I said, hydrochloroquine for malaria, all the malarial patients. Okay, that is a generalized medicine. But when you come to personalized medicine, we know that certain drugs in cancer they're only manufactured on demand. So when a specific person is there, he is known susceptible to cancer, then a specific medicine will be manufactured for that person at that moment, and then that medicine will be given to him. That is personalized medicine. And this is not new. Now people are saying that we are giving a specific medicine for personalized medicine for people, but this is what Indian medicine system used to follow. So if you have gone through the Ayurvedic system, which we used to have earlier, it is very specific. It is personalized medicine. Even for the same person who is having cold, the same medicine will not be given to the same person, to different persons. Okay, each person will get different medicine on basis after deciding the three factors which we call them as Vata, Kapha, and Pitta. Okay, so these are the three variations what people used to have in the Ayurvedic medicine, and if you can understand all these three, then these three will be different for three different persons. Okay? And a specific medicine will be prepared immediately and then will be given. So the same concept now has come to the people. Now, based on your disease, based on your genome, a medicine will be devised and developed. Okay. and then it will begin. And that is what we call as personalized medicine at this moment. And at this particular condition, Japan is the first country which is actually having more personalized medicine. Okay, more personalized medicine. So now we'll see what is that. So this technology has actually made available personalized medicine to people. And why this is possible? Because your human genome sequencing has come down to nearly $1,000. That means we can get sequence at 75,000. This is what $1,000 says that. But in US, for $1,000, a human genome can be sequenced. That is, it is afforded by a normal person. That is what it is said. And it's very quick and reliable. Okay, it is very quick and reliable. So what is that we are doing? So DNA, that is a blueprint of life, is actually the molecules. We know that it encodes genetic instructions. Okay. So it encodes genetic instructions. So this blueprint is actually taken by. So the technology required here is sample preparation, data processing, and low throughput and short release. So this is what we have. And this fourth generation is actually giving all this thing. And the other advantage what we have is label free. We don't label anything now in this fourth generation. And we are actually sequencing long sequences, long reads, what we say. Okay, minimum requirement of materials. Okay, so here you can see here, as we have seen with the previous thing, we do not prepare a particular sample. We don't require data processing algorithms. It is not a low throughput result. It is actually generating a high throughput data. We are generating long reads, and it's again a low cost. So these are the advantages of this. At the same time, it's label free. Okay? Label free. We are not even labeling them. So this is actually based on the technology which is originated from Coulter counter and ion channels. Okay. So we have seen previously that when we pass the DNA through the protein nanopore, ions channels, that is voltage is generated, and that voltage is actually used. Same way we have this here, but here our technology is different here. Okay, so the whole fourth generation, there are two types of methodologies which are used here. Whether you use a biological material or use a chemical material. Okay, whether you use a biological material or a chemical material. So those nanopores are actually made up of solid state film or embedded in a biological. Solid state is a chemical nature of a material, and biological membrane is a biological nature of material. Okay. So when the ion passes through these membranes, so what happens here is there is cis and trans compartments which are there, and electrolytes are there. So when these electrolytes are actually allowing this DNA to pass through this membrane and cis and trans compartment, there is a voltage fluctuation which is generated, and that voltage fluctuation is actually useful for detection of the nucleotide. Okay, so here it is. You can see here, like your electrophoresis chamber, it is very simple. We have electrodes which are immersed in each chamber, and a bias voltage is passed, and electrolyte ions pass through the solution through the pores, and that gives you a signal, and that signal is activated. So the pore is blocked by analyte, and the negatively charged DNA is added to the chamber, blocking the current going to the chamber. Okay, so this will actually give you. So how we do this? So I'll just uh, show you it on a whiteboard. Okay. Again, you can see here. So we have a DNA. Okay. You have a nanopore material here with the pore here. And here, instead of this, in this, we have a chamber. So you have electrodes here. Okay. So we have an electrodes here, which are there. Okay. So in this electrodes, this is actually there is a solution here all around. So when current is passed through this electrodes, there's this electrolytes which is present in the chamber will actually allow the DNA to pass through this pore. Okay, and this DNA when is coming outside, it will generate ion or current. So here the chamber will be like this. Okay, so this is a so the DNA will pass through like this, it come here, and then it will move like this, then in this direction. So in cis and trans positions, when it moves in these directions, electricity is generated. And this electricity or ion, which is actually here. So this is the meaning of this. Okay. So there are electrodes. So here you can see here. So there is a reservoir or a chamber which is of cis and trans. So I've shown you how a cis and trans compartments look. Okay. In this cis and trans compartments, the electrolytes will pass along with the DNA. So once they pass, okay, once they pass, through that they generate electricity. So electrolytes which are immersed in each chamber will generate electricity, and these electrolyte ions pass through the electrophoretically, resulting in ion current signal. This ion current signal is actually taken into consideration. Okay, and this will give you. So as said, there are two types of material which are there. One is biological nanopore technology material, and other solid. So we'll tomorrow in the next class we'll see how is biological nanopore how biological nanopore technology is used for solid state nanopore technology is used for sequencing the genome. Okay. So is it clear till here about the third generation sequencing technology and nanopore technology?
Sir.
Yes, sir.
Okay. So the next class will continue with this, how a nanopore, solid nanopore material or biological nanopore material are used for genome sequencing. Okay then. Right. Thank you. And have a
Thank you,
nice day.
sir.
Thank you, sir.
Where did we stop? The last class? Anyone? Balaji?
Sir, we were done with nanopore.
Yeah, which generation? Nanopore. Because nanopore is present in
Third generation, sir.
Third
Third
generation.
generation.
yeah.
Third generation nanopore.
We completed third generation, sir.
And did we start fourth generation? Did we start? I think we start fourth generation also.
Some part, only some part we did, sir, in the previous class.
Okay. So what we have seen in third generation is that we have two technologies which are available for generation of sequencing data. First one is nothing but single molecule real time sequencing approach, and second one is Oxford nanopore technology. Well, both of them were real time. We don't have any methodology to first record and then later reduce the sequence. Here it is, the sequence is directly given as an output here. Okay, and we've seen what is there in nanopore technology. Coming to nanopore technology, we have seen that actually there is the DNA sequence first will be separated into two, first template read and complement read. Then one sequence will be taken and that will be passed through the nanopore. Once that is passing through the nanopore, an ion fluctuation will be generated, and that ion fluctuation is actually equivalent to the voltage, and so that is recorded as a one nucleotide. So each nucleotide has a different voltage fluctuation, and those will be recorded. Okay. With this, you are able to now have the sequence data of DNA using nanopore. Okay, so with this, the nanopore has advanced a little bit technology, and that is what we have learned in the last session. That is, there is a fourth generation sequencing technology, something but a biological nanopore sequencing approach and a solid state nanopore sequencing approach. Here, what happened is people started thinking what different levels of DNA sequence can be done based on different materials which are used in the nanopore technology. Okay? And whatever material which was used in nanopore, one type was with the biological nanopore biological material, and the second was with the chemical material. And whatever was made with biological material, there was biological nanopore sequencing approaches, and whatever were made with chemical materials, they were known as solid state nanopore sequencing. Okay, so now we'll see what are they. Okay, so now fourth generation nanopore technology has become much more advanced and it is able to sequence the data personalized medicine. And that is what I said. What we discussed in the last week, that personalized medicine has become a new way of treating diseases. Where I try to differentiate between the personalized medicine and general medicine. So the
Sir, your voice is not clear, sir.
voice is not clear.
Now it's okay, sir.
Okay. So whenever we have a personalized medicine, I mean, a general medicine is given to all the persons. The same medicine is given to all the persons. Then that is known as generalized medicine. And if a specific medicine is especially synthesized and given to a person, then we call it as a personalized medicine. And fourth generation sequencing has allowed us to more, go towards personalized medicine. And now it has become here. And now it is possible because each and every person's genome can be sequenced at $1,000, which is a psychic thing of spending the money in US. Whenever something is near to $1,000 or below $1,000 in US, it's known that it is a common man affordable object. Okay, when something is near $1,000, that means they assume that that technology has reached to the common people, and it can, as the same way, be achieved, and it can reach all the people all over. That is the same. And even this has now the sequencing technologies in the US has come down near to $1,000, and India has also come down near to 1,50,000. Okay, 1,50,000. It is almost, I mean, double. Here it is $2,000 for us. Okay, if you want to sequence it, and in US it is just $1,000 per genome. Okay, and there are many companies in Hyderabad also which give you your human genome sequence to you. Okay, but the only thing is people are not there to understand after sequencing your own genome. Okay, people are not there in this area. Okay, so one important opportunity. So all of you can come online now. On your videos, all of you. All of you on your videos. Akash, Anjali, Manudepak, Srividya. Okay, so I was stressing on a point here, one important point, that there is a availability of the genome data being sequenced at a lower price. Okay, $1,000 and $2,000. When something is reached to the common people, then what happens? It is commonly used, isn't it? So genome sequencing technology is available now to sequence data, but to analyze the data and to understand the data, people are not there. Is it clear? Now, all of you have a chance to make an opportunity in this field. That means in future, it would be like this. I mean, how many of you have gone through before going to a doctor, maybe you have done some tests and then taking that report to the doctor and asking for an test, isn't it? This is a general way. Maybe in the near future, the doctor will prescribe the test in 3 different ways. Molecular test, okay, biochemical test, that's what we are going to lab, and a bioinformatics analysis. That means now people are already in a procedure to store your stem cells, isn't it? As soon as we are born, they're trying to deposit the stem cell bank. That's the routine approach. Now what is called? Now the second routine approach, what will come is, as soon as the baby is born, the genome will be sequenced first. Maybe it will become a mandate after 20 years. At this moment, it's not there. But after 20 years, it will become a mandate. Okay. So once your genome is sequenced, once your DNA is available, so what should be done? The report is also analyzed and given at the moment the baby is born. So whenever you go to the doctor will ask you to just go through the report once again and see at this point of age and time, is this person has a susceptibility of getting this disease according to your genome. This is the depth people will soon see as a reality. Okay, and this is what people used to do in Ayurveda. People used to observe 3 points: Vata, Pitta, and Kapha. We don't know what are they. We don't have the complete knowledge. So if you can go to the doctor, I mean, an Ayurvedic doctor, he will explain. But at this moment, Indian organizations, all group consultorium has done the test, and they have found out that they are linked to SNPs. Vata, Kapha, and Pitta are linked to SNPs. SNP, single nucleotide polymorphism. Okay. Between us, each and every person you are seeing on the screen or in your family, there is a single nucleotide difference between you and your family member. And that nucleotide difference is actually contributing to the difference between you. And that can be the difference in the disease also. So the same medicine cannot be given. And that was what identified by the Ayurvedic system. And that's why no single medicine was given when you go to Ayurvedic doctor. Within the family also, if two persons are going in the same feeling that we are susceptible to cold. But the medicines will be given, two different medicines will be given to two different, the same, I mean, same members in the family. Okay? Because your genetic composition is different. Maybe you're of the same family, but composition is different. That's why you require a different medicine. Got it? The same thing will happen in the future. First, a person, genome will be taken, genome will be analyzed by a bioinformatician, and then report will be generated by him or her, and that report will be taken to the doctor, and the doctor will start understanding. So doctor's role is much more. Now he has to understand bioinformatics. Now he has to understand biochemical analysis test. Now he has to understand genetic counseling. Okay, genetics. Then he has to describe the medicine. So he has to go through all these things, and then the doctor has to do the medicine. But before that, there is a lot of space under that people for analyzing the genome data, more and more people are required. Even not only that, more and more people are required for creating and maintaining the database. So where do you keep your genome? Assume that each and every person will have his personalized genome in a pen drive. Okay, and you will also see that your genome data is stored somewhere in a private property, like you're storing all your files in a cloud at this moment. Maybe you store your genome data in a cloud. Okay, and you have to give the access to the persons. But who will maintain the data? If you lose a copy of person's genome in the pen drive, who will maintain the data? So there is a second opportunity you can think that this is another job opportunity. First thing is genome analysis. Second thing is maintaining the genome data. Maybe you can make a business also from that. Okay, two options are there. Persons who are running the mathematics analysis for human genome, and persons who are running the database management with private features for all the genome. Two important businesses, two important shortage areas. And third one, I said that human genome data is too large, 3.2 billion base pairs, isn't it? Now, can you just compare your genome with other genome? I'll say that that is not possible with whatever computers we are using now. I'm using the latest and high-end computer at this year, but this is also not enough for analyzing your genome. Comparing two human genomes, I'm not talking about comparing all the genomes. Two human genomes we can't compare. Okay, imagine now, what is another opportunity we have now? The next opportunity what we have here is again making a device or making algorithms to compare all these genomes. in a single code. That means if I want to make a comparison of all your 21 genomes, whatever people you are there in the class, 31 genomes, I require a high-end computation system to do that. Even that is not available. Okay, so there are three important opportunities you have to remember at this moment, where you can make a career, or where can make a business from these things. Okay, at this point I'm saying these are the three things you can think of making a career in your life. In future, these will be very common things. Is it clear? Huh? Okay, so now we move into the next generation technologies in the class. Okay, so now this $1,000 has brought all these things, and these are the opportunities what are listed now. Okay, so what is that which has made this fourth generation easy? Preparation of sample is very easy. Data processing is very easy. Algorithms, complicated algorithms are not there. It's low, low in cost, and it is generating actually higher. So whatever I read in these other technologies for sample preparation, these are all the drawbacks of the previous one. And the advantage is that it has become sample preparation is very less, it's label free, it's high throughput, and it requires a minimum sample. All these are the advantages of the fourth generation sequencing technology. Okay, so what did they do here? Again, simple thing. I think I even gave you this. Okay, so there's a chamber with this made up of crisscross, cis and trans compartments. Okay, so in that chamber we have a very good just 1 minute. Just 1 minute. I'll take. I've shown you how fourth generation sequencing technology is actually sequencing your data. Okay, so there is a chamber. There is a chamber like this. Okay, and that chamber is actually having a tube like this, and these are cis and trans compartments which are there. Okay, with electrodes here. Okay, so when DNA is passing like this. You can see the red part is DNA. It is passing in cis and trans way through the compartments. Okay. As so, while this is passing like this, what is happening here is the flow will generate ions, and this will generate the current. So this is what is happening here. So here you can see that there are pores either made up of solid state film or biological membrane. Okay, and whenever there is a chamber which is there, okay, and reservoir with cis and trans compartments of electrolytes. Okay, so the electrolytes are immersed in each chamber, bias voltage is passed, and ions are passed through the pores outwardly, resulting in the current signal. And this current signal is actually give you a voltage, and that will be detected by the Coulter counter ion channels, okay, and converted to voltage. So this is what happens. So this is the chamber, but these are the electrodes, okay, and this is your DNA passing through the cis and trans membranes. When voltage is electrodes to this electrodes, voltage is applied, the DNA will pass through the cis and trans, and it will finally give you a DNA. Okay, so when it's passing, ion voltage will be generated, and ions will be collected, and voltage will be generated, and this voltage is actually detected by Coulter chamber, okay, ion detector, and this will give you the DNA signals. Okay, Coulter counter and so to have this material which is there are two types of materials which are used for generating this electricity and DNA sequence. What are they? First one is actually a biological nanopore technology and membrane. Second one is known as a solid state nanopore technology and membrane. Okay, so biological nanopores are nothing but the proteins, the transmembrane protein channels, which we already know, which you have seen in cells, the bilipid layer, or the polymer films, or liposomes. All these are the biological nanopore materials which can be made into channels in the chamber. Okay, the major advantage of this is that reproducibility, size, structure, and combination of the way we can combine and make it. That is the advantage. Okay, so what are the biological nanopore materials? Bilipid layer, polymer films, or liposomes. Okay, all of these are the biological nanopores which are used in this biological nanopore technology to sequence the DNA. Okay, so what are the common examples we see? So the first material which was actually found and used is alpha hemolysin, okay, which is actually for reading information from 4 nucleotides. So this can read 4 nucleotides. Bacteriophage 529 and Oxford nanopore technology. So these are the biological nanopores. So what is that? It's used in Oxford nanopore technology. It's a protein. Okay? It's a protein nanopore. So what is hemolysin? So hemolysin is actually an alpha toxin, which is actually exotoxin, which is secreted by bacteria Staphylococcus aureus. Okay, it's an exotoxin secreted by bacteria, human pathogen Staphylococcus aureus. Okay, so these are the materials what we have. Now, what is the advantage of this alpha? Alpha hemolysin size is inner diameter of alpha, and size of the single stranded DNA are pretty close in size. Okay, so alpha hemolysin and single stranded DNA both are same. So this is actually a very simple. So pretty close in size that aids in identifying the nucleotides using ionic color. So whatever single stranded nucleotide is there, that can be easily identified by this hemolysin. So when this single stranded DNA is actually passing through this hemolysin, the actually generate a current which is different. So identify nucleotides using ionic current in the nanotube. It is a positive for all of their interaction analysis. Okay, so this material has made easy for us to use this nanotube. And what is the limitation here? So this limitation here is its pore size. So alpha hemolysin and ssDNA are equal size almost. And if that is the case, sometimes if the nucleotide sequence is a little bit longer, it cannot go. Okay, it is not able to differentiate that nucleotide. So that is the limitation. Okay, so this is the limitation. So once again, we'll see what is biological nanopore technology here. Okay, so biological nanopores are nothing but the bilipid layers, polymer films of ribosomes. Okay, the advantage with them is the size, reproducibility, and ability to modify them. Okay, and the best examples which are already used in making biological nanopore technology for sequencing: hemolysin, MSPA, bacteriophage, fine nucleotide, and Oxford nanopore technology, protein. Okay. Now what is hemolysin? We'll take one example here, biological nanotechnology. And hemolysin is actually a toxin which is released by a bacterium Staphylococcus aureus. Okay. And the alpha hemolysin is actually very equal in size to a single stranded DNA. Okay. So whatever single stranded DNA is moving to this hemolysin can produce ionic current, which is easily differentiating all the nucleotides. But the only limitation is both of the sizes are very similar. Okay. So Balaji,
Sir,
so how we can use fourth generation sequencing technology for sequencing the DNA strand? Yes. Gurudev,
Sir,
can you tell?
sir,
Can you tell how we can use fourth generation sequencing technology?
sir, I didn't get you, sir.
How do we use fourth generation sequencing technology to sequence a genome or a DNA strand? Padmini? Padmini? Okay, carefully listen again. Okay, so we'll take a DNA, a chamber which is having compartments with cis and trans, where two electrodes are present at two different ends. Okay. So when you pass on the DNA strand will move through that cis and trans compartments and generate ionic current. This ionic current is actually detected by Coulter ion chamber. Okay, and that is easily in a position to differentiate that whatever nucleotide ionic strength is generated, and each nucleotide will have a different ionic strength, and that will be detected as a nucleotide. Okay, in this way we can easily detect it. So the same thing, what we are doing here is we have used two different materials. The first one what I explained here is nothing but your biological nanopore material. Those biological nanopore materials can be your proteins, okay, your alpha hemolysin, MSPA, bacteriophage, pinene, and another one which is there. Okay, so these materials are used. And among them, whatever I explained now is nothing but your alpha hemolysis. Alpha hemolysis is nothing but a toxin which is actually secreted by Staphylococcus aureus. Okay, and that is equivalent to the size of alpha hemolysis. So now what is happening here? This material is actually made into that whatever compartments which are there using this alpha hemolysis. Okay, so the single stranded nucleotide strand can go through this alpha hemolysis nanoprotein and give you a nucleotide with by differentiating. So when the output is there, you can easily differentiate all the four nucleotides separately. So in this way, you can get all the nucleotides. So that is the advantage. And the disadvantage is almost alpha nucleotide, single stranded nucleotides, and alpha hemolysin are the same size. So sometimes there can be a problem when you're using a larger single stranded DNA sequence. Okay, larger single stranded DNA sequence. So this is what we are trying to learn in biological nanopore technology. So we have a second technology which is now here is chemical nanopore technology, and we'll meet again after 5 minutes. Okay, at 10:43 we'll meet and we'll complete that chemical nanopore technology. Is it clear?
Yes, sir.
Okay, welcome back to the session here. Now, all of you here?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So in the previous session, you have seen about biological nanopore technology. Now we'll see about the solid state or chemical nanopore technology. Okay. So here, instead of biological materials available to us, like protein bilayers, biolipid layer, or alpha hemolysin, or bacteriophage, phytone, okay, instead of them, we are using here chemical substances or chemical materials to make this nanopore, and that is known as solid state nanopore technology. Okay, solid state nanopore technology. So whatever nanopores which are here, they are made with the nanomaterial. Okay, and that nanomaterial is actually used. So what is the advantage here? So biological nanopore is just a biological material, we're going to use that. But if you want to make some small changes in the features, you can't make it. So this biological, the advantage of the solid state nanopore with this biological nanopore is that we have a thermal stability, chemical stability, and mechanical stability. I think you know what are these three. Okay. We have a thermal stability, that is, it is heat resistant. It is stable even at temperatures. Chemical stability, it is resistant to certain chemicals. Most of the chemicals they cannot harm this. Okay, it is resistant to strength, stability. So thermal, chemical, and mechanical stability are And what is the most important thing here? Size. Nanopore means the material should be of nanopore size. So through this nanopore, the DNA can pass. So the size is also an advantage. So thermal, chemical, mechanical, and size adjustable. You can adjust your size based on your DNA. Okay, that is it. So all these made this solid state material as an advantage over the biological material, nanopore material. Now, what were the things which were used here? Okay, what were the things which were used? That is silica, okay, and silica oxide. These are the two materials which are used in making this membrane, solid state nanopore technology. Okay. Another one is aluminum oxide. Okay, aluminum oxide membranes. So these are the three different versions of nanopores which are used. And what it gives is, while using silica, more chemical stability is there and mechanical stress is reduced. While using aluminum oxide, more electrical performance was there. That is, we said that DNA is moving through this nanopore, electrical pulse will be generated. And that has improved a lot when you use aluminum oxide. Okay, now what it gives advantage? The major advantage what it gave was we were able to sequence the DNA. Okay, that we are able to detect the proteins, then we were able to diagnose the disease. So these were the advantages what we got while using a solid state nanopore technology. Okay, so these were the advantages what we had. So what are the other advantages what we have? Low noise while translocating the DNA. That means there's a little bit of disturbance when the DNA is actually passing through the different chambers of the DNA have a nanopore material. Okay, and we used to get a good ratio, signal to noise ratio. Okay, and there are different materials which are used here. Single membrane layers, hybrid of both biological and solid. Okay, and so hybrid of biological and solid. So these are the different solid state nanopore technologies which are used. Okay, so this is what which is there with solid state nanopore. So the advantage here is we can easily diagnose the disease, identify the proteins, and sequence the DNA. Okay, and we were able to easily make it using it, and we get a definitely chemical heat and mechanical stability with this. Okay, so the materials which are used are silicon oxide and aluminum oxide. Okay, one gave a chemical stability, another one gave electrical improved performance. This has reduced the noise during translocation, and the signal ratio is high. And two types of metals are available for this: single layer membranes, hybrid or biological solid state. Okay, so these are the different fourth generation. Now we'll see what are the fifth generation. Okay, so the point we have to look at here is the fifth generation sequencing technologies is that technology actually there is a little bit of lower accuracy when we sequence ultra short DNA sequences. Okay, ultra short DNA sequences. So till now, till this generation, first to fourth, we have concentrated on sequencing large genome or large DNA sequences. But we forgot about the short DNA sequences which can be used. Okay? So these short DNA sequences were a problem. So this fifth generation sequence has actually offered a material to sequence this ultra short DNA sequences. Okay, ultra short DNA sequences, and accuracy has improved a lot. Till fourth generation accuracy was a little bit low, but from fifth generation the accuracy was very high. Okay, and nanopore technology again. So it has, you can see the nanopore technology has changed its state, and DNA sequencing has changed along with the nanopore. Now there are two approaches here. First one is high fidelity nanopore sequencing of ultra short DNA targets, and second one is ultra sensitive nanopore sequencing of cell-free tumor DNA. These are the two different approaches which are available. Okay, so high fidelity nanopore sequencing of ultra short DNA targets is a nanopore based sequencing strategy. Okay, let us first sectorize. So first the DNA is sectorized, then amplified via ruling sector, and then it produces repeats of the sequences. Okay, so this is what is followed here. First is sectorizing, then amplifying via amplification, okay, and then produce concatenate sequences which are shortly joined. Okay, so these are sequenced then on a billion platform. So with this procedure, we try to get the ultra short read sequence, and then we take it to the machine, that is a MiniOIN platform. What is that? Previously we have seen here MiniOIN in third generation. Okay, nanopore sequencing technology. There we have seen that there is a machine which is known as MiniOIN. So first we circularize, amplify, and then make a concatenate repeats, and then we give this sample to the MiniOIN platform, and this will actually give a sequence which is ultra short sequence which is sequence. At the same time, the accuracy is high when compared to the previous one. Okay, so we are able to sequence sequences which are less than 100 base pairs. Okay, with this we are able to sequence the sequences which are 100 base pairs. So this is the approach which was followed. First thing is we take your DNA, we amplify the DNA via rolling circular amplification. Then we make repeats. These repeats are sampled to MiniOIN platform machine, and then there it sequences and gives you a data which is less than 100 base pairs and more accurate than the previous one. This is about high fidelity nanopore sequencing, okay, of ultra short DNA target. Now we have a second approach. Okay, that is ultra sensitive nanopore sequencing of cell-free tumor DNA. Cell-free tumor DNA. Okay, so what is this here? How many of you have heard about cell-free tumor DNA?
No,
How
sir.
many of you heard?
No, sir.
No. So,
No, sir. We didn't get across it.
so I think you have an idea about cancer in general.
Yes, sir.
Okay. So whenever is a cancer or tumor, the cancer cells or tumor cells are spread throughout the body, isn't it?
Yes, sir. In malignant stages.
Okay. So when they pass, actually they release a DNA which is known as cell-free tumor DNA. So from the cell, they are freed, and this tumor DNA is there in the blood. Okay. And now we are actually concentrating on detecting this cell-free tumor DNA. If we can detect the cell-free tumor DNA, you can know in what stage the cancer is, and then you can give the treatment. And these are actually the first initial molecules that when the cancer starts spreading from that position where it is there. Okay, so that's why people started concentrating on this. So this cell-free tumor DNA is actually identified here, and the same methodology is followed here. We take the cell-free tumor DNA, we perform a rolling circular amplification, we capture the circular molecules, and then sequence them. Okay, using MiniOIN. So what is actually happening here? When you do this, you actually capture this cell-free tumor DNA, short DNA sequences, and then you can say that, okay, this is a tumor DNA and this can cause cancer. Okay, so this is how we're able to identify. Now, what were the advantages with this? I think you have heard about the single nucleotide variants. I told you about single nucleotide polymorphism now, S and S. That there is a nucleotide difference between two individuals. If those two individuals are varying, then we call them as single nucleotide variants. Okay, these single nucleotide variants can be identified with this 5th generation sequencing technology. Okay, so in short, what we have learned is we have learned that first generation sequencing technologies like Sanger chain domination method, Maxwell Gilbert sequencing method. Second generation sequencing technologies like Roche 454 sequencing, Iotoro sequencing, Illumina Solixa sequencing, and ABI solid sequencing. And coming to third generation, we have learned about single molecule real-time or SMRT sequencing approach and Oxford Nanopore technology sequencing. And coming to fourth generation technology, we have seen that biological nanopore sequencing technology and solid state nanopore sequencing technologies are there. And coming to the fifth generation sequencing technologies, we have seen that high fidelity nanopore sequencing of ultra short DNA and cyclic and ultra sensitive nanopore sequencing of cell-free human DNA. So this is what we have learned now. Okay, so with this we have covered what are the different generations of sequencing technologies which are available for sequencing a genome. Maybe a long fragments or maybe short fragments. Any of them can be sequenced using these 5 generations of sequencing technologies. Until date, there are 5 generations of sequencing technologies which are available to us. Is it clear?
Yes, sir.
Okay. So here and there we have seen that there are few technologies which are actually developed or devised into machines, and this is actually the technology which was driven for high throughput genome sequencing. Okay, high throughput genome sequencing. So in the next class, we'll see what is high throughput genome sequencing. And for time being, go through these notes, whatever PPTs I've uploaded, and on Friday, we'll discuss what are actually high throughput genome sequencing and how these next generation sequencing technologies and previously genome sequencing technologies strategies helped in high throughput genome sequencing. Okay, so on Friday we'll meet with the next topic of high throughput genome sequencing. I hope I'm clear till this next generation sequencing technologies. So any doubts?
No, sir.
Yes or no?
No, sir.
No? Okay. So Friday we'll have a session after we complete the high throughput genome sequencing technologies, and then we'll clear whatever session we have, because are people aware of your first speed exam? When is your first speed exam?
24th, sir.
24. That means we have another 1 week. Okay. So for till Friday, this Friday you try to complete whatever you have learned till now. Any doubts in them, you cover them. Okay. So that you'll be prepared for your exam. Is it clear?
Yes, sir.
Okay, then we'll meet again on the next class on Friday. Okay. Bye. Thank you and have a nice day.
Thank you, sir.